Welcome to Lesson 7.05 on proteins and nucleic acids. Our essential question for today is, what are proteins and nucleic acids and what do they do in my body? In our goals, we want to be able to illustrate the formation of carbohydrates, lipids, proteins, and nucleic acids, identify the relationship between amino acids and proteins, and identify the relationship between nucleotides and nucleic acids. Here's our mastery map. Organic molecules, some examples of those are amino acids, which form proteins through peptide bonds, and nucleotides, which form nucleic acids. So what do proteins do? Well, they provide you with nutrition. You can eat protein in food. That's how you get your protein that you need for your body. They provide structure to parts of the body, including hair, bones, and muscle. They also act as enzymes. You probably talked a lot about enzymes in biology. These speed up chemical reactions within cells. They also transport nutrient molecules through the blood. They possess properties of toxins to protect various species of animals. So some animals use proteins as toxins to escape predators. They also send signals through the body to do various tasks. So a lot of the tasks that your body carries out uses proteins for that. Proteins are polymers. We talked about polymers 2 lessons ago. Those are really, really big biological molecules that are composed of repeating units of something. We call those a monomer. Specifically for proteins, they are composed of amino acids. Amino acids, here's the basic structure of one right here. They have an amino group. They have a carboxylic acid group, they have a hydrogen, and then they have what's called an R group. This is different. So there's 20 total amino acids. Each one has something different right here where I have an R. This R group is typically what gives the amino acid its specific properties. So there's 4 main types of R groups in amino acids. So there's 20 total amino acids. And just like I said on the previous slide, every amino acid has this amino group, this carboxyl group, the hydrogen, and then this group is what's different. So some of them will have a non-polar R group. These contain mostly carbon and hydrogen in their chemical structure. Isoleucine is an example of that, so it has the amino group, the carboxylic acid, but if you look at that R group, it's mostly hydrogen and carbon. Polar R groups will contain electronegative atoms, so you'll typically have oxygens, nitrogens, fluorines. Serine is an example of that. It has that OH hydroxyl group that makes it electronegative and makes it polar. Aromatic R groups will contain alternating double bonds in their chemical structure, so you'll see these ring-like structures here, like in phenylalanine. You can see the alternating double and single bonds right here. And then you have your charged amino acids. They'll have a charge. All amino acids will usually display a charge in their carboxylic acid and in their amino group, but in the R group you'll actually see a negative sign right here or a positive sign. So those are your charged amino acids. Like I said, amino acids are the monomers that form the polymer known as proteins. So if you think back to that Lego example, proteins would be like your giant Lego structure. Amino acids would be like the individual Legos that you put together to form it. So here's an amino acid, here's an amino acid. You put them together, and the bond that they're connected through is called a peptide bond. So that's this right here. That's what connects two amino acids in a protein. It's sometimes called an amide bond. I've heard it referred to more as a peptide bond though. So the monomer is amino acids, the polymer is protein. The amino acids are connected by peptide bonds. There's basically 4 levels to protein shape. As you guys probably learned in biology, the shape of a protein is what gives it its function. Primary structure, that's just the way the amino acids are connected in a line. So you might have proline, then serine, then leucine, then isoleucine. So that's just the way they're attached here. Secondary structure, you have hydrogen bonding between the backbone, so between the amino and carboxylic acid backbone. Tertiary structure involves interactions between those R groups. And then quaternary structure, this is only if you have multiple polypeptide chains within your protein, and that's the interaction between different polypeptide chains. What I want you to know here, though, is that proteins have very large complex structures that are determined by interactions within the molecules. Next thing we want to talk about are nucleic acids like DNA and RNA. They are made up of building blocks known as nucleotides. Nucleotides have the basic structure that's shown right here. They have a phosphate group, a sugar, and then a base. And the base could be one of four things. It could be one of the two purines, adenine and guanine, or it could be one of the two pyrimidines, cytosine or thymine. So what you see here where it says base, it's one of these four things. There's also uracil as well. That's a fifth base that you could have that's only found in RNA. But that's what you'll see right here. It's different among the different nucleotides. And there's only, like with amino acids, there's 20 different ones. With nucleotides, there's only 5 different ones. Uracil's not pictured. So nucleotides form the polymers known as nucleic acids. Those are things like DNA and RNA. And that's the same reaction that we talked about with amino acids forming proteins. It's that condensation reaction. You're putting these two monomers together and you're losing a water molecule. The bond that connects them is called a phosphodiester bond. So when you put these two nucleotides together to form a nucleic acid, they're connected by what's called a phosphodiester bond. Specifically in DNA, DNA is made up of two large nucleic acid strands that come together to form the double helical structure that you see in DNA. And so this backbone right here, that's the phosphate group and the sugar. And then this would be the base that we talked about, either the A, the T, the C, or the G, or the U if you're talking about RNA. These bases actually hydrogen bond to each other, so you'll have hydrogen bonds in between each of these. That's what forms it into this double helix right here. This information in DNA, that's what has the instructions for which proteins to make and how to make them. So your DNA actually has all the instructions for making your proteins. That's how that all fits together. So it goes DNA, then RNA, then protein. That's the central dogma that you probably learned about in biology.